Rabies. Introduction. Rabies is an acute, fulminant, fatal encephalitis. It is called the madman disease, which has instilled terror in human society. The reason is that, with rare exceptions, all of the people who were bitten by a rabid animal got rabies. Rabies is an important zoonotic infection in which man is the dead end of infection and hence doesn't play any role in its spread to a new host. In most of the developing countries, dogs are the principal reservoir of rabies, canine rabies, whereas sylvatic rabies involve animals such as foxes, raccoons, cats, and coyotes. Rabies has been recognized from very ancient times. The word rabies is derived from the Latin word rabidus, which means mad. It is an epidemic disease. History. 1885 – Development of human rabies vaccine. 1903 – Diagnosis of Negri bodies. 1940s – Use of rabies vaccine for dogs. 1954 – Addition of rabies immunoglobulin. 1958 – Rabies virus grown in cultured cells. 1959 – Development of fluorescent antibody test. Causative agent. Rabies virus causes rabies infection. It belongs to the family Rhabdoviridae and genus Lyssa-virus. In Greek, Lyssa means rabies. Rabies virus is bullet-shaped, size about 180 into 75 nanometer, with one end rounded or conical and the other planar or concave. Genome is negative sense single-stranded RNA. It is nucleocapsid in nature. Two layers, the matrix layer and the outer envelope, cover the genome. Matrix is made up of M protein. Outer envelope is made up of lipid bilayer similar to the plasma membrane. External envelope has spike-like projections. It is made up of glycoproteins. Spikes are responsible for the pathogenic property of the virus. RNA-dependent RNA polymerase is responsible for genome replication. L and P proteins control its activity. Rabies viruses of man and animals all over the world appears to be of a single antigenic type. Antigens of rabies viruses are G protein, M protein, N protein, heme agglutinin. Chemical composition. Chemically, the rabies virus is composed of 4% RNA, 67% protein, 26% lipid, and 3% carbohydrate. Resistance. Rabies virus is highly resistant to cold, dryness, and decay. The virus is highly thermolabile with a half-life of approximately 4 hours at 40 degrees Celsius and 35 seconds at 60 degrees Celsius. The virus cannot withstand pH less than 4 or more than 10. It is also susceptible to oxidizing agents, most organic solvents, surface-acting agents, and quaternary ammonium compounds. Proteolytic enzymes, ultraviolet rays, and X-rays rapidly inactivate the virus. Soaps and detergents are effective against the rabies virus because of their lipid eliminating property, which destroys the outer envelope of the virus. Replication. The rabies virus binds to the cellular receptor and enters the cell via receptor-mediated endocytosis. The viral membrane fuses with the membrane of the vesicle, releasing the viral nucleocapsid. This structure comprises of negative sense RNA coated with nucleocapsid and a small number of L and P proteins which catalyze RNA replication. Negative sense RNA is copied into 5 subgenomic mRNA by L and P proteins. The N, P, M, and L mRNAs are translated by free cytoplasmic mRNA. gmRNA is translated by ribosomes bound to the endoplasmic reticulum. Newly synthesized P, N, and L proteins are involved in RNA replication. This process begins with positive sense RNA synthesis. Positive sense RNA of the host serves as a template. Some of the negative sense RNA enters into viral protein synthesis. gmRNA transcribes and synthesizes glycoproteins. G proteins travel to the plasma membrane. Progeny nucleocapsid and M proteins are transported to the adjacent area of plasma membrane. Assembly takes place and new viruses are released through budding process and infect new cells. Clinical manifestation. Incubation period is about 30 to 60 days. Clinical features in man. The clinical features in man include hydrophobia, lockjaw, encephalitis, hysteria, acute polyneuritis, and poliomyelitis. Clinical features in dog. The clinical features in dog are change in behavior of dog, change in tone of bark, change in feeding habit, off feed, conception of abnormal objects, fever, vomiting, excessive salivation, paralysis of lower jaw, restlessness, convalescence, and paralysis leading to death. Clinical features in cat. Clinical features in cat are aggressiveness, great sensitivity to touch, profuse salivation, attempt to attack dog or even man. Pathogenesis. The virus present in the saliva of a rabid animal is deposited at the biting site. The virus attacks via spikes to the nicotinic acetylcholine receptors of host tissue cells. The virus appears to multiply in the muscles, connective tissues, or nerves at the site of deposition. It penetrates the nerve endings either immediately or after a varying interval and travels into the exoplasm towards the spinal cord and brain. The movement of the virus in the axons is passive at a speed of about 3 mm per hour. The virus then multiplies extensively in the brain tissue, causing symptoms of encephalitis. Characteristic inclusion bodies called Negri bodies form at the site of viral replication in the brain, but the cells are not lysed. The virus spreads outward from the brain via the nerves to various body tissues, notably the salivary glands, eye, and fatty tissue under the skin, as well as heart and other vital organs. The immune response of the host probably plays an important role in pathogenesis, since viral antigens are expressed on the surface of the infected cells. Freshly isolated viruses are called street virus. If this virus is passed through various cell lines, it is converted into fixed virus. It is used for vaccination. Laboratory diagnosis: specimen, saliva or sputum, skin biopsy, hair follicle, cerebrospinal fluid, blood, corneal swab, urine, and brain post-mortem form the specimens for diagnosis of rabies. Laboratory test: Negri body examination, mouse inoculation, complement fixation test, polymerase chain reaction, hemagglutination test, passive hemagglutination test, electron microscopy, enzyme-linked immunosorbent assay, fluorescent antibody test, serum virus neutralization test, counter-immunoelectrophoresis, immunoperoxidase test, hemagglutination inhibition test, passive diffusion, and tissue culture techniques. Negri body examination. Negri bodies are intracytoplasmic inclusion bodies. Seller's staining procedure is followed to demonstrate the presence of Negri bodies. First, the brain is cut open to expose the hippocampus region. A small piece is cut and placed on a filter paper with cut surface facing upwards. The filter paper is then placed on a glass slide. The cut surface is slightly sponged with the edge of the filter paper to remove blood. A clean microscopic slide is pressed on the tissue piece. While the smear is wet, it is flooded with the working stain and allowed to stain for 2 to 3 seconds and then washed with water, air dried and examined under 100x. Observation: nerve cells, blue cytoplasm and dark blue nucleus. Stroma appear as pink, erythrocytes copper colored, and negri bodies magenta to dark red with dark blue or black inner granules. Prevention and treatment. This section was categorized into three. All three carry equal importance and one should not be given undue importance or neglected at the cost of other two components. These components are management of wound, post-exposure immunization and pre-exposure immunization. Management of wounds. Since the rabies virus enters the human body through a bite or scratch, it is imperative to remove as much as saliva with soap. After the removal with soap, any quaternary ammonium compound, for example 1% cetrimonium bromide, may be applied as an antiseptic along with anti-rabies serum. Post-exposure immunization. Because of the long incubation period, it is possible to institute prophylactic post-exposure immunization. Immunization must be started at the earliest to ensure that the individual will be protected before the rabies virus invades the central nervous system. Two types of agents are employed to confer immunity to an individual who has been exposed to the rabies virus: anti-rabies serum or rabies immunoglobulin and anti-rabies vaccine. Anti-rabies serum provides passive immunity in the form of ready-made anti-rabies antibodies to tide over the initial phase of infection. Anti-rabies vaccines that are available in India are Semple's sheep brain vaccine, human diploid cell vaccine HDCV, primary chick embryo cell vaccine PCECV, purified vero cell rabies vaccine PVRV. Site of vaccination. The ideal site for vaccination is the anterior abdominal wall. This area offers enough space to accommodate 10 injections at 10 different sites and cause least discomfort to the patient. Dose schedule. A 6-dose schedule spread over a period of 3 months is recommended. It is also called Essen schedule, proposed by International Conference on Rabies held at Essen, Germany. Days of vaccination are 1st, 3rd, 7th, 14th, 30th and 90th days. Pre-exposure immunization. There are no vaccines available for mass pre-exposure vaccination. The pre-exposure prophylaxis hence is recommended for definite group of individuals who because of their profession or hobby are at a higher risk of getting exposed to rabies virus. High risk group for rabies: laboratory staff handling virus and infected materials, veterinarians, animal handlers and catchers, wildlife officers, quarantine officers, naturalists in epizootic areas, travelers from rabies-free areas to rabies endemic areas. Control of rabies. Any strategy for control of rabies in developing countries shall have the following component: epidemiological surveillance, mass vaccination, dog population management, and community participation.